Hello friends, and welcome in my channel Easy Life Science. Here I am talking about some important questions, their answers with detailed explanations for SET exam subject Life Science part 10. So please subscribe my channel Easy Life Science, like my video and share it to your friends, and don't forget to press the bell icon so you'll get the notification of my newly uploaded video. So let's start. Question first, all of the following can be chemically isolated except. Some options are given here. Reading the first option, enzyme substrate complex. The enzyme substrate complex can be chemically isolated by increasing a concentration of a inhibitor. Option second, enzyme inhibitor complex. It can be chemically isolated increasing a substrate concentration. But when the enzymes binds with the substrate through a covalent interactions, then it cannot chemically isolated. So the answer for this question is option third. Then the next question, severe form of a galactosemia is. The galactosemia, it is a disease in which there is a high level of a galactose in the blood. This is an inherited disorder prevent your body from breaking down the sugar galactose, causing it to build up the toxic levels in your blood. Galactosemia caused by a deficiency of an enzyme galactose 1-phosphate uridyl transferase enzyme. So option first is the answer of this question. The galactosemia is a genetic deficiency of a uridyl transferase that exchange a galactose 1-phosphate for a glucose on UDP glucose. So option first is the answer of this question. Here is the pathway for a galactose metabolism. The galactose is a monosaccharide. It gets break down into galactose 1-phosphate through an enzyme glucokinase. But because of the enzyme or because of a lacking enzyme GALT, that is galactose 1-phosphate uridyl transferase, the galactose 1-phosphate does not get converted into a UDP glucose. So it leads to a increasing level of galactose in a blood. The affected organs are like a brain, a liver and a bone. So option first is the answer of this question. Then the next question, ionophores are. The ionophores are chemicals that forms the pores in the plasma membrane and allow ions to pass through. Ionophores are lipophilic chelating agents that transport cations across the phospholipid bilayer and complexes with monovalent cations such as sodium ions and calcium ions and cross the cell membrane and enter the cell via ion transport system in exchange for hydrogen ions and potassium ions. Two types of ionophores are there. One is a channel former and another one is a carrier. The channel formers are usually large proteins. They are incorporating themselves into a cell membrane and creating a polar tube across the cell membrane and enabling ions pass through it. Whereas the carriers are known as mobile ion carriers, which are small molecules that physically shield the ion from surrounding environment and facilitate its movement across the plasma membrane. So answer of question number 5th is option third. A chemical that forms pores in a plasma membrane and allow the ions to pass through. Then next question, the two ATS ribosomal subunits remains united with each other due to high concentration of which of the following ions? The divalent magnesium ions are essential for several aspects of a protein synthesis, including neutralizing the charge on a ribosomal RNA for allow it to correctly fold and play an important role in subunit association. The ribosome ATS is made up of 60S and 40S subunits. They get united with each other with the help of magnesium ion. So answer of this question is option third. Then the next question, which of the following occurs in a meiosis but not in a mitosis? If you compare a meiosis and a mitosis, the pairing of a homologous chromosome at a metaphase plate, pairing of a homologous chromosome at a metaphase plate, that is a synapsis, it occurs only in a meiosis, not in a mitosis. Other three options, that is attachment of a spindle fiber to a kinetochore, it occurs in a mitosis as well. Replication of a DNA prior to a start of cell division, it appears in a mitosis, and a separation of a sister chromatid at an anaphase, this also occurs in a mitosis, but a pairing of a homologous chromosome at the metaphase plate, it occurs in meiosis only. So option second is the answer of this question. Here in this image, there are some differences in between a meiosis and a mitosis. The mitosis produces 2 daughter cells, whereas the meiosis produces 4 daughter cells. In a mitosis, daughter cells are diploid, that is a 2n. In a meiosis, daughter cells are haploid, that is a n. In a mitosis, daughter cells are genetically identical. In a meiosis, the daughter cells are genetically different. The growth, repair, and replacement of old damaged cells in a higher organism form of asexual reproduction in a lower organism. For example, in a prokaryotes, it is a characteristics of the mitosis, whereas a formation of sex cells or gametes, which are necessary for reproduction in higher organisms, formed in a meiosis. The mitosis helps in multiplication during a early period of growth, whereas the meiosis helps to maintain a genetic diversity in a population. The mitosis occurs all organisms except viruses. Meiosis occur in animal, plants, and fungi. Here are some another differentiating characteristics are there. You can take a screenshot of it and read further. So moving to the next question. The amount of microbial mass produced from a nutrient can be expressed quantitatively as, it is expressed as a growth yield. The cell count, that means a measurement of a viable cells, means there is no any direct relationship to the microbial mass. Balanced growth and a dilution rate, these are incorrect option. So answer of this question is option second. The next question, what is RNA primers used for a bacteriophage T7 replicating DNA? At time of replication of a bacteriophage T7, the primer used is a PPP, AACC, PPP, ACCC. So second option is the answer of this question. Then the next question, the following features of a genetic code should be observed, and you have to read it and choose a correct option from it. First, most amino acids have more than one codons. Yes, it is correct, because the asparagine, aspartic acid, cysteine, glutamine, glutamic acid, histidine, lysine, phenylalanine, and tyrosine, these amino acids are coded by 2 codons each, whereas alanine, glycine, and proline, these amino acids are coded by 3 codons each. Methionine and tryptophan, these amino acids are coded by 1 codon each. So the option first is a correct option, and this is about a degeneracy of a codon. Most amino acids have more than 1 codon. This is correct. Then next, second, 3 codons signal termination of a polypeptide synthesis, that is the stop codon UAA. Yes, it is a stop codon or a termination codon, or it is also called as a nonsense codon because this codon does not code for any amino acid. And the UAA, it is known as a ochrei, UAG umber, and UAG as a opal, and UGA as a umber. So these are stop codons and not coding any amino acids. Then third, one codon signals initiation of a polypeptide synthesis, the start codon AUG. Yes, it is correct because the AUG is a initiation codon in a prokaryotic organism. The AUG codes for N-formyl methionine. In case of eukaryotes, it codes for methionine, and this AUG on a mRNA is identified by a initiation factor first. So this is also true. So all the sentences are true. So the option 4 is the answer of this question. Then the next question, the gal operon has 3 cystons. The gal operon is a prokaryotic operon which encodes for enzymes which are necessary for a galactose metabolism. Glucose and lactose cannot act as an inducer because they don't have a binding site for attaching the repressor protein. The gal operon is controlled by calf cyclic AMP complex, similarly to that of the lactose operon. And the galactose operon having 3 cystons, that is gal E, gal T, and gal K. So option first is the answer of this question. Then the next question, name the RNA polymer first and type of a gene it transcribes. The RNA polymer first is responsible for transcription of a ribosomal RNA. Okay. Eukaryotic transcription, here there is a role of 3 different RNA polymerases: RNA polymerase first, RNA polymerase second, and RNA polymerase third. But in case of prokaryotes, single RNA polymerase is there and it transcribes all the 3 types of RNAs. Looking for a eukaryotic transcription, the RNA polymerases are of the 3 different types: RNA polymerase first, second, and third. The RNA polymerase first, it transcribes ribosomal genes 20S, 80S, and 5.S. These genes or these ribosomal genes are transcribed by a RNA polymerase first. The RNA polymerase second transcribed mRNA genes, whereas RNA polymerase third transcribed 4S and 5S tRNA. So 20, 80, and 5.S, these ribosomal genes are transcribed by RNA polymerase first. So option first is the answer of this question. Here option first is the answer of this question. 80S, 5.S, and 20S RNA or 20S ribosomal RNA are transcribed by RNA polymerase first. So option first is the answer of this question. Then moving to the next question. Stem cell factor is produced mainly by. The stem cell factor is nothing but a glycosylation protein, and it is produced mainly by a bone marrow stromal cells. So option third is the answer of this question. Stem cell factor is a glycosylation protein produced by bone marrow stromal cells. Then next question, the direction of a flagellar rotation is modulated by the signaling molecule. The signaling molecule, that is CHEY, and this CHEY get phosphorylated and rotate the flagella in a counterclockwise direction and the cell to a clockwise direction. So option third is the answer of this question, that is a CHEY. This is a signaling molecule for a rotation of a flagella. Then the next, familiar cancer is caused by. This is a straightforward question, and the familiar cancer is because of a mutation in a germ-line cells only. So option second is the answer of this question. Then the next question, HIV attacks human immunodeficiency virus. It has spikes, that is a GP120, and these spikes are an attachment organ for attaching to the CD4 plus cell or a T helper cells. So the option third, that is a T helper cells or a CD4 plus cell, is the answer for the question number 16. Then the next question, phytochrome action influenced by long day can be substituted by. It can be substituted by the phytochrome gibberellin. Other options are there, auxin, antioxidant, and a cytokinin. Before a gibberellin is a hormone which is responsible for cell elongation and internodal growth. Whereas the auxins plays a crucial role in controlling plant growth and yield by regulating embryo development, seedling development, tropism, and apical dominance. The auxin, this phytochrome, is produced in the apices of plants and help to control the growth. The option second is antioxidant. The antioxidant means the chemicals or compounds that acts against the auxin or inhibit the physiological functions of the auxin. Antioxidants like para-chlorophenoxyisobutyric acid is PCIB, para-chlorophenoxyisobutyric acid PCIB. This is an antioxidant, and this hormone competes with auxin to its binding site. Other example of antioxidant is NPA, that is 1N-naphthylphthalic acid, and another example that is TIBA, that is 2,3,5-triiodobenzoic acid, which blocks auxin transport. By blocking the transport of auxin, they prevent the action of the auxin. The option third is a cytokinin. It is responsible for cell division. So for particular this question, answer is a gibberellin. Then the next question, animals that exhibit discoidal cleavage pattern in their meroblastic egg. A discoidal cleavage, here a cleavage from a disc-shaped primer cells 1 to 2, 2 to 4, and such cleavage is known as a discoidal cleavage, and it is shown by a fish, reptiles, and birds. Fish, reptiles, and blood. Option 4 is the answer of this question. Then next question, plastochron is the term that speaks about. The plastochron is defined as the time intervals between two successive recurring events during a growth of plants. Shoot development such as starting from a leaf initiation, and this index is given by Erickson and Michelini. So it speaks about a leaf development. So fourth option is the answer of this question. Then the next question, the pattern of a leaf arrangement where a leaf form a node departs in a pair at a right angle in a successive nodes is called as. Four options are there. The arrangement of leaves on a stem is known as a phyllotaxy. Leaves can be classified as either alternate, spiral, opposite, or folded. Plants with alternate and spiral leaf arrangements have only one leaf per node. The alternate phyllotaxy It is an arrangement of leaves in which one leaf is present at each node. All the leaves lies in the same plane. For example, hibiscus. The second is opposite. Opposite phyllotoxy in which the two leaves are present on each node. The leaves are opposite to each other. The opposite phyllotoxy is classified into two types: opposite decussate and opposite superimposed. The opposite decussate, in this type the leaves of two adjacent nodes lie at 90 degrees to each other. For example, calotis. Opposite superimposed, in this type two leaves of adjacent nodes lie at the same plane. For example, eugenia. And further phyllotaxy, in this type three or more than three leaves are present around the single node. It is also known as a verticillate type. So for this question, answer is opposite decussate phyllotaxy. Here in this image, here a type of a phyllotaxy: opposite, alternate and furrowed. Alternate, arrangement of leaves in which one leaf is present at each node. Opposite, in this type of a phyllotaxy two leaves are present on each node. Two leaves are present on each node. In alternate, one leaf is present in one node. And furrowed, more than three leaves are present at one node. So answer for this question is option 4, that is opposite decussate phyllotaxy. Then next, during non-cyclic electron transport, phosphorylation occurs at. A non-cyclic electron transport during a photophosphorylation, here is a non-cyclic photophosphorylation. The excited electron form a photosystem second with a high energy stored in it get transferred to a number of electron transport carriers in a photosystem or in a electron transport chain. Pheophytin, so plastoquinone to cytochrome B6, cytochrome B6 or cytochrome F, plastocyanine and plastocyanine to then photosystem first to a pyridoxine reducing substance, then to pyridoxine and then to a NADP, where the NADP get converted into a NADPH2. In this case, this electron never come back to a photosystem second. It is accepted by a NADP, so electron does not get cycled. So it is a Z scheme photophosphorylation or a non-cyclic photophosphorylation, and in this photosphorylation, phosphorylation occurs at one step where the electron moves from a cytochrome B6 to a cytochrome F. Here the ATP molecule is formed from a ADP and one inorganic phosphate. In a non-cyclic photophosphorylation, at a one step there is a formation of ATP, but in case of cyclic one, here is a cyclic photophosphorylation, photosystem first only involved in it. The excited electron with a high energy stored in it get transferred to a pyridoxine reducing substance, then to pyridoxine, then to cytochrome B6. Here the ADP get converted to a ATP, then a cytochrome B6 to a cytochrome F, another molecule of ATP form, then plastocyanine and plastocyanine to a it reversed back to a photosystem first. So electron is cycled. But in this cyclic photophosphorylation, at two stages there is a formation of a ATP molecule. But in case of non-cyclic, at a one stage there is a ATP formation. So question is asked on this. Question is asked on a non-cyclic electron transport, photophosphorylation occurs at one place, cytochrome B6 to a cytochrome F. Then the next question, glycolytic reactions of a respiration occurs in eukaryotes and in prokaryotes. The EMP pathway or a glycolysis, that is glucose get converted into a pyruvic acid, and this pathway of a glycolysis, formation of a glucose, formation of a pyruvic acid from a glucose is carried out in a cytosol. But in case of eukaryotes, the TC cycle occurs in a mitochondria, and also on a mitochondria the respiration takes place through a electron transport chain. The perioxysomes are responsible for a breakdown of the hydrogen peroxide or reactive oxygen bases. Glycosome where the glycolytic cycle occurs. So the answer of this question is the option second, that is a cytosol. In a cytosol there is a glycolytic cycle, glycolytic pathway, not cycle. Then the next question, which one of the following is an antioxidant? The antioxidant is a compound, or it is the chemical, or it is a hormone that acts against a oxygen or inhibit a physiological function of a oxygen. Oxygens are like indole acetic acid, indole butyric acid, and a 2,4-D. These are the oxygens. But the antioxidants like a PCIB or a para-chlorophenoxyisobutyric acid, this hormone competes with oxygen to its binding site. Other examples of antioxidants are NPA, that is a 1N naphthyl acid, and a TIBA, that is a 2,3,5-triisobenzoic acid, which blocks oxygen transport, and by blocking a transport of oxygen they prevent the action of oxygen. So antioxidant, that is a 2,3,5-triisobenzoic acid, that is a TIBA. TIBA is a antioxidant, triisobenzoic acid. Then next question, classical experiment by understanding the nature of a circadian rhythm in a plant were carried out by. The circadian rhythm, the scientist Jean-Jacques Dortus de Marian, Jean-Jacques Dortus de Marian, he was a French astronomer. He gave the idea about a circadian rhythm, who placed a mimosa plant in a dark room and observed that the plant continued to unfold its leaves in a morning and close them in a evening. It showed a circadian rhythm. The plants have an internal timekeeper known as a circadian clock that anticipates environmental cues such as a light and a temperature. So answer for this question is a Jacques Dortus de Marian. Read the other options like Charles Darwin. The Charles Darwin, he is known for his contribution to the evolutionary biology. His propositions that all the species of life have descended from a common ancestor is now generally accepted and considered a fundamental concept in science. Then the William Pfeffer. The William Pfeffer was the pioneer of modern plant physiology. His scientific interest included the thermostatic and photonestic movements of the flowers. And the fourth option, Francis Darwin. The Francis Darwin, he was the third son of Charles Darwin. So answer of this question is option C, third, that is D. Marian. The next question, blood flow that is directed through the lungs and back to the heart is called, it is called as a pulmonary circulation. It is a division of a circulatory system in all vertebrates. The circuit begins with a oxygenated blood returned from a body to the right atrium of the heart, where it pumps out from the right ventricle to the lungs. In the lungs, the blood is oxygenated and returned to the left atrium of the heart and complete the circuit. This is a pulmonary circulation. The other division of a circulatory system is the systematic circulation that begins with receiving the oxygenated blood from the pulmonary circulation into left atrium. From atrium, the oxygenated blood enters the left ventricle where it is pumped out to the rest of body, returning as a deoxygenated blood back to the pulmonary circulation. The blood vessels of a pulmonary circulation are pulmonary arteries and pulmonary veins. So option third is the answer of this question. So next question, which of the following is an incorrect association? So read one by one. The first, that is anoxia. The anoxia is a condition when body or a brain completely loses its oxygen supply. Anoxia is usually a result of a hypoxia. This means that a part of a body does not have enough oxygen. Hypoxia can be a consequence of many conditions, includes a low oxygen at high altitudes, significant blood loss, carbon monoxide and other gas poisonings, and breathing difficulty that lowers the oxygen supply like asthma. Anoxia is harmful to brain. After about 4 to 5 minutes without oxygen, your brain can become permanently damaged. So deficiency of O2, that is anoxia. It is true. Now second, dyspnea, labored breathing. Dyspnea, it is a medical term for a shortening of a breath, overexertion and spending a time at a high altitude are responsible for dyspnea. So it is a labored breathing. It is also true. Apnea, this is a condition when you stop breathing while asleep or have almost no airflow at time of sleep. So this is incorrect. Excessively high breathing rate, it is not correct. Also read the fourth options. Pleurisy, it is an inflammation of a tissue that lines the lungs and a chest cavity. Infections such as flu and certain medications can cause a pleurisy. So it is also true. Inflammation of a pleura. So option third is incorrect. So answer of this question is option third. Then the next question, which of the following statement is correct with respect to a kidney function regulation? Read one by one. During summer, when body loses a lot of water by evaporation, the release of ADH is suppressed. Yes, it is correct. Sorry, not. During summer, when the body loses a lot of water by evaporation, release of ADH is stimulated. So it is incorrect option. When someone drinks a lot of water, ADH release is suppressed. Yes, it is true. But also read the another options. Exposure to a cold temperature stimulates ADH release. No, it is stimulate, does not stimulate, it suppress it. So it is also incorrect. An increase in a glomerular blood flow stimulates a formation of angiotensin second. It is also incorrect option. So option second is the answer for this question. The next question, a hormone involved in regulating basal metabolism is. The thyroid hormone regulates the basal metabolic rates of the body. Tetraiodothyronine, often known as a thyroxine or a T4, and triiodothyronine, that is a T3, are the two hormones that the thyroid gland can secrete. Iodine level in a body control the synthesis of these hormones, and these are involved in regulating the basal metabolism. So answer of this question is a thyroid hormone. Read other options also. Insulin, This is secreted by beta cells of a pancreas. It controls a glucose level in a blood. Then the gastrin. Gastrin is a peptide hormone that stimulates the secretion of gastric acid by parietal cells of the stomach and aids in a gastric motility. And this gastrin hormone is released by the G cells in the pyrolic antrum of the stomach, duodenum and a pancreas. And a glucagon, it is secreted by alpha cells of a pancreas. It raises the concentration of glucose and fatty acid in a bloodstream and is considered to be main catabolic hormone of the body. So option third is the answer of this question, that is a thyroid hormone responsible for regulation of a basal metabolism. Then next question, number of types of a gamete produced by a heterogamous individuals is. Heterogamous individual means a human being or males. They produce two types of gamete, X and Y. So option first is the answer, heterogamous. If the question is homogamous, then the homogamous produces only one type of a gamete, that is for example females produce X gamete only. So answer for this question is option first. Then next question, the gene formed by the joining the DNA segments from different sources are called as. This term is known as chimeric genes. Chimeric genes form a combination of a portion of two or more coding sequences. So option first is the answer of this question. Thank you friends, thank you for watching my video. If you like my video, please subscribe my channel, share it to your friends, and don't forget to press the bell icon so you'll get the notification of my newly uploaded video. Thank you once again.